The overall goal of the following experiment is to generate a possible zebrafish model of polycystic kidney disease. This is achieved by injecting morpholino against wnt5a in transgenic WT1-BGFP zebrafish embryos at the one-cell stage. Next, a rescue experiment is performed by microinjection of mouse wnt5a mRNA with the wnt5a morpholino. Then, the injected embryos are anesthetized and embedded at 48 and 72 hours post-fertilization in order to monitor kidney structure and cyst formation after wnt5a knockdown. The results show that wnt5a knockdown causes pronephric kidney cyst formation in transgenic WT1-BGFP zebrafish based on fluorescence microscopy of pronephric kidney structures.
The implications of this technique extend toward building a model of cystic kidney disease, because the transparency of the zebrafish embryo, coupled with the use of kidney-specific fluorescence protein transgenic, allow a direct view of kidney structures in a live organism.
This method can provide insight into wnt5a knockdown causing zebrafish kidney cyst formation. It can also be applied to test many other genes that are suspected to cause renal cyst formation. This protocol is simple and less time-consuming.
After designing and synthesizing morpholinos according to the manufacturer's instructions, add high-grade, sterile water to the glass bottles to thoroughly